Алло. Аишки. Хорошо. Давай скорую. Но это не дело, у тебя давление прыгает то 170, то 120. Так нельзя. Значит, увезет в больницу. Ничего тебе не делать, раз тебе давление скачет. Да, вызывай дозу. Да. Давай, пока.
Что там, Тань?
Был утром, я выпил таблетку 120, потом опять 170, сейчас опять померил. То есть промежуток 2 часа прошлось.
Ну, чуть добавлять, не держит, значит, один этот пока. И у него вообще эффект развивается не мгновенно.
Угу. Сначала у него снизился от него.
От него или само?
Угу.
А она что, ее качает? Там может быть микроинсульт.
Я ей тоже так сказала.
А что она собралась делать?
Ну, эта дядька сейчас кричит, я вызову скорую. Она говорит, нет, не надо мне никого.
Дед не работает, что ли?
Вторую смену.
Так, у нас с тобой по крови что?
Сначала температура.
По-моему, нет. Нет, мы вот эти все форменные элементы, все до эритроцитов дошли. Не говорили, да. До эритроцитов дошли. Вот composition of blood function, вот это мы все сделали. А теперь эритроциты. Good morning, dear student. Good morning. We are continue the lecture to theme blood physiology. The theme today, physiology of erythrocytes. An erythrocyte structure and function. An erythrocyte of a person has no core, mitochondrial lysosomes. It is highly specialized structure, which main function is to transfer oxygen from lungs to tissue and carbon dioxide from tissue to lungs. Gases are transferred in connection with hemoglobin, which locates in erythrocytes. The main source of energy in erythrocytes is glucose. Erythrocytes have the biconcave form that allows them to pass through capillaries. The erythrocytes lives in a vascular bed for 120 days. On its membrane, there are C-align aside, which give is the negative charge. On the membrane, there are also group and rhesus factor antigens. Several mechanisms of these, of an old erythrocytes, are divided. 1. The erythrocyte is phagocyted by macrophages. Next, intravascular, hemolysis, and next in a spleen, a liver, and bone marrow. Next question, regulation of erythropoiesis. The erythrogenesis is a process of formation of erythrocytes in bone marrow. This process includes the following phases. Colony-forming cell erythrocytes. Next, 1 pro-erythroblast. Next, 2 basophilic erythroblast of the first ooza. Next, 4 basophilic erythroblast of the second ooza. Next, A polychromatophilic erythroblast of the first ooza. Next, 16 of polychromatophilous erythroblast of the second ooza. Next, 32 polychromatophilous normoblastis. Next, geniculation of normoblast. Next, 32 reticulocytes and and 32 erythrocytes. The erythrogenesis before formation of erythrocyte takes 5 days. The erythrogenesis at the person proceeds in erythroblastic islands of marrow. Macrophages of erythroid cell are part of these islands. Macrophages phagocytes. The nucleus which are pushed off from normoblasts provide the erythroblast with ferritin and plastic substances, secrete erythropoietin, strengthen growth promoting substances in islands. Reticulocytes have marrow into blood and turn into erythrocytes within a day. Normal reticulocytes make 5 to 10% of all erythrocytes circulation in blood. Decrease of quantity of erythrocytes in blood is anemia, increase is erythrocytosis. The main regulator of erythrogenesis is erythropoietin. They develop in kidneys, macrophages of marrow, Kupffer cell, and hepatocytes, and amount of them depends on the level of oxygen in blood. Sex hormones, androgens, increase sensitivity of cell of erythroid row to erythropoietin and strengthen erythrogenesis. Hemoglobin. Hemoglobin and its combinations. Hemoglobin in the hemoprotein containing iron. 1 liter of muscular blood contains 104 to 175 grams. Feminine blood has 123 to 153 grams of hemoglobin. Hemoglobin is placed in erythrocytes and makes them red. Combination of hemoglobin with oxygen is oxyhemoglobin. With carbon dioxide is carbohemoglobin. With carbon monoxide is carboxyhemoglobin. In all these combinations, iron in hemoglobin is divalent. At action of the strong oxidizers, iron in hemoglobin turns into tervalent. The methemoglobin is formed by it. The oxyhemoglobin, which has given oxygen, called reduced or dioxyhemoglobin. Erythrocyte sedimentation rate, ASR. Mechanism, ASR. If the blood deprived of an opportunity to buy cartilage, placed in a capillary with a diameter commensurable with a diameter of an erythrocyte, and part a capillary upright, formed elements will be divided into plasma, and formed elements, the majority of which is erythrocyte. ASR is estimated on plasma column higher over a column of formed elements. Normal ASR makes for men from 2 to 10 millimeter per hour, for women from 2 to 20 millimeter per hour. Mechanism ACR. The erythrocyte appeared in blood from red marrow has a negative superficial charge. In the course of life, in the blood, cause protein, for example, fibrinogen, products of inflammation, disintegration of tumors and toxins, contact with surface of erythrocyte. As these molecules shield a charge of erythrocytes and ASR accelerated. Thus, ASL is an integrated indicator of blood composition. So, next question, physiology of leukocytes and thrombocytes, hemolysis. Questions, leukocytes, quantity, leukogram. Next, regulation of granulocytopoiesis and monocytopoiesis. Next, Lymphocytes, regulation of lymphocytopoiesis. Next question, thrombocytes, quantity, function, and to end types and mechanism of hemolysis. Let's start from leukocytes. These cells formed powerful blood and tissue barriers against bacterial, viral, and parasitic infections and stimulate tissue regeneration. Adult human blood contains from 4 by 10 to the 9 degrees leukocytes. Their increase up to 10 to the 9 degrees in leukocytes is leukocytosis, and decrease of less than 4 to 10 the 9 degrees is leukopenia. We distinguish physiological and reactive leukocytosis. By physiological, we mean food, myogenic, emotional. Reactive leukocytosis develops as a result of inflammatory process and infection, disease. It strengthens increased formation of leukocytes in the bone marrow. Leukopenia occurs only with pathology associated with inhibition, granulocytopoiesis. So, leukogram. Let's start from basophilic. Basophilic ranged from 0 to 1% of the total number of leukocytes. Function of basophilic are maintenance of blood flow in small vessels by secreting heparin into them. So, secretion of hemoattractant for eosinophils. Next, activation of the growth of neocapillaries. Next, protection of the intestine, skin, and mucous membranes when infected with worms and mites, and phagocytosis. Next, form eosinophils. From 1 to 5% of the total number of leukocytes. Functions are protect the body against parasitic infections, inactivate biologically active compounds formed during allergic reactions, have phagocytic and bactericidal activity. Next form, neutrophils. Neutrophils ranged from 45 to 75% of the total number of leukocytes. Function, phagocytosis of damaged cells, bactericidal secretion and stimulated tissue regeneration materials, and destruction of penetrating into the body of pathogenous microbial infections. 30 to 16 seconds after contact of neutrophil with bacterium, there arises flash metabolism with the formation of hydrogen peroxide, superoxide ions, and hydroxyl radicals. They oxidize halogens bacteria membranes. Both neutrophils secrete lysozyme, cations, proteins, lactoferrin, hydrolase, which destroy phagocytosis bacteria. Neutrophils is infected tissues can also form an extracellular bactericidal trap. End group monocyte or macrophagocyte is about from 8 to 10%. They provide phagocytosis defense against microbial infection, toxic effect on the parasites, participation in the immune response and inflammation, tissue regeneration by stimulating them in fibroblast, endothelial, vascular, and smooth muscle cells. Regulation of hemopoiesis and phagocytosis of old and dematted blood cells. Next question, regulation of granulocytopoiesis and monocytopoiesis. Granulocytopoiesis and monocytes occur in the bone marrow. Colony-stimulated factor, which is secreted by monocytes, macrophages, and T lymphocytes, stimulates neutrophilpoiesis granulocyte. Colonies and lactoferrin secreted by neutrophils oppress neutrophilpoiesis. Eosinophilpoiesis stimulate interleukin 5, 3, interleukins 4 and 3 stimulate the products of basophils. Monocytopoiesis stimulates monocyte colony stimulating factor. Attention, dear student, it is mind regulator of monocytopoiesis. Prostaglandins 8, alpha and beta, interferon, then secrete monocytes inhibit. Lymphocytes. Regulation of lymphocytes. Lymphocytes make up 28 to 45%, while the following blood has only about 2% of all lymphocytes. Cell live from a few hours to many years. Subpopulation of T and B lymphocytes are divided by the functional properties. Lymphocyte functions, provide immune responses, regulate the activity of other cells in immunological reactions, regulate growth, differentiation, and tissue regeneration by contact, interaction, and secretion of cytokines. Multiplication and differentiation of lymphocytes before exposure of antigen lymphopoiesis occurs in the bone marrow and thymus. The process of lymphocyte differentiation after exposure to an antigen is called immunopoiesis. Regulation of T and B lymphopoiesis in the bone marrow is performed by stromal cells. They inhibit by lymphopoiesis interleukin 1 alpha, tumor necrosis factor, interferon gamma, steroids. Next question, thrombocytes. Thrombocytes as adults are 150 to 320 10 to the 9 power per liter. Thrombocytes are involved in blood clotting, reduced integrity of damaged blood vessels, exhibit antiparasitic activity. In thrombocytes, we identify several areas that perform different functions: peripheral, Solgel, and intracellular organelles. In the peripheral zone, there are clotting factors, enzymes, and receptors required for activation, adhesion, and aggregation of thrombocytes. In the Solgel zone, there is thrombocytes contractile apparatus. In the circulation blood, there is up to 17% of platelets, and about 30% is in the spleen. Platelet lifetime is from 7 to 10 days. Intracellular organelles zone includes granules which contain ADH, calcium ions, serotonin, epinephrine, platelet derived growth factor, and lysosomal enzymes. Thrombocytes are produced in the bone marrow. thrombocytopoiesis is activated by thrombopoietin. Formation of thrombopoietin is stimulated by a decrease of platelet thrombocytopenia. So, next question. Hemolysis, types and mechanism hemolysis. Hemolysis is the destruction of the red blood cell membrane and hemoglobin arrival to the environment. Depending on factors damaging the red blood cell membrane, we distinguish hemolysis chemical. Membrane is divided by acid or alkalis. Thermic hemolysis. Membrane is divided if the temperature drops below 0 degrees or rises above 40 degrees Celsius. Biological hemolysis. Membrane is divided by position of insect, reptiles. Immunohemolysis. Red blood cell stick together and a complementary group antigens and destroyed by antibodies. And osmotic hemolysis. Erythrocytes are destroyed in hyperosmotic sodium chloride concentration of less than 0.85% medium. So, dear students, the lection